Okay, we are ready to go. I am Eva Garcia Muntion. I am the chair of BioAqua. I'm here with my colleague Mattia, the chair of EUROESTOP. We are organizing this webinar, the two actions together, which makes it also very
Eva, we cannot hear. You are muted.
Sorry. I was welcoming everybody. Thank you. I was welcoming everybody. I am very happy that you are all here. This is a webinar organized together by two actions, EURESTOP and BioAqua. Mattia is here with me, is the chair of EURESTOP. I'm the chair of BioAqua. We are very happy that this collaboration is being done. It makes it even more exciting. We have three speakers with different topics, all of them more or less related to something that we could describe as new chemical solutions and pathogens and pollutants affecting farming, the environment and society, particularly in water environment. And that's all from my side. Mattia, do you want to introduce the topics and maybe the first speaker?
Yeah, thank you, Eva, and thanks to all the people connecting. I see that there is a very large participation, which is very, very nice. We are really happy about it. The speakers have been selected in order to have complementary topics between the two constructions. Some of them are also involved in both the constructions, and we believe that this may be a nice starting point for further collaborations and also to explore a topic from a different angle. The webinar is organized in an easy way. We let the speaker to speak. We collect in the meantime your questions in the chat. So if you have a question that you can ask later, just write it in the chat, possibly also specifying the speaker who you want to be answering. If you have general questions for all the speakers, is fine as well. After all the talks, we will have a 20 minutes question and answer session. So we will read your questions to the speakers and they will respond to your questions. So I think we can start with the first speaker, who is Simona Bartkova. And the talk is The Wonderful World of Droplets. Oh, I was reading in your slide. Let me take again. Diving into the possibilities of droplet-based microfluidics for antimicrobial resistance and microplastic pollution research. Please, Simona, can you share the screen and upload your presentation?
Yeah, I hope if you can.
Yeah, see your screen. Try to go in presentation mode.
Yeah, sorry, it's up here. Should be.
Yeah, we see also, yeah, but we see also your next slide and notes. So if you can remove this.
You need to swap. We are seeing the presentation mode.
Yes, I see that. Let's see. Technical. Maybe I should then instead share.
You can go in the slideshow option. Can you share again? So
yeah.
We can.
Maybe I can directly share if I share directly.
You have a button. There is a an option on the top that says swap. Yes. I think if you swap that. Oh, okay. Now we see it properly.
Yeah. Can you try to go to the second to see if it is working? Yeah. Perfect.
Okay. Nice.
Yeah. Thank you.
All right. When I go back. Yeah. All right. So I will start. Thank you very much, Mattia, for the introduction. I'm involved in both cost actions, so both EUROESTOP and BioAqua. And my name is Simona Bartkova. I'm a researcher at the Department of Chemistry and Biotechnology at the Tallinn University of Technology, which is in Estonia. So before we dive into the world of droplets, I do have a question just to wake up everybody. So I'll be talking partly about microplastic. So let's see if you can find the raised hand. Do you think that micropollution is an urgent threat? I do see some hands up. Very nice. It's fine if you don't agree, or if you cannot find the button, then it's also fine. All right, good. So a lot of people at least think that is the urgent problem, and I agree. So if we look further, if we look at microplastic pollution as a threat to aquaculture, well, the research shows that mostly actually farmed fish are typically containing more microplastic than wild-caught fish. And this can come from many different things in the feeds, with plastic packaging, from the cages, from different things, many different things. And they can lead to all in the aquaculture and then aquaculture products. Okay. And this microplastic can then accumulate and it can lead to many ecotoxicological effects. So those are just some that can happen in the fish. But that is not only the problem here. So if you look further as well, why it's a threat as well to humans. So we like to eat fish, and this is not the only way we get microplastic. It is everywhere now, even in the air that we breathe. And it can enter the human placenta, so it can lead to harmful toxins, harmful for pregnancy, can cross the blood-brain barrier and cause brain injuries and neurotoxicity. It has also been found in our blood, okay, in arteries, affect cardiovascular health, and it can also accumulate in the liver, and it has been shown that it can cause inflammation and damage. So it's not a positive future for microplastic. But microplastic pollution has shown as well to be able to enhance antimicrobial resistance. Okay, but how does it do so? Okay, how does it enhance antimicrobial resistance? Well, if we look, so it's kind of simple story about aggregation biofilms. So usually if you have one single bacterium, they don't like antibiotics, but they can meet other bacteria and they can form together these aggregates, or on a surface, it can be biofilm, and produce extracellular polymeric substance. So this kind of makes them protected against antibiotics. And how the microplastic then comes in, well, it can provide this perfect nesting ground. Okay, we produce, and this number is probably even higher now. Okay, we produce 400 million tons of global plastic waste. So this is just the waste per year. So during weathering, it can break down and can alter as well, both physically and chemically, okay, to what we kind of call microplastic or nanoplastic. But this can then attract pollutants. Okay, this can be metals, that can be pesticides, antibiotics, bacteria, everything. It's basically like a magnet. Okay, and it means over time, and it now has a unique name. It can produce this ecosystem that is called a plastisphere. Okay. All right, so this is kind of a complex system, but if you go back to the droplets here, how do they come in all of this? Okay, what role does droplet-based microfluidics play? Well, it is a versatile tool, okay, and is based on manipulating immiscible fluids. It means fluids that don't mix. So it can be, for example, oil and water. And usually what is done is that you have a droplet generation microfluidic chip. So these chips where you can then have oil flowing plus what we call surfactant that makes the droplet stable, and then you have a water-based sample. However, you can also mix and match, have a oil in water or oil in water in oil in many different things you can combine to make things a bit more complex. But the main thing is that you should think of each water in oil droplet as one single test tube. Okay, so this means you can have thousands and thousands of experiments sort of in one single test tube instead of having thousands and thousands of test tubes on your like bench or whatever in your lab. Okay, so this means the positive sides of droplet-based microfluidics. It's chemical separation. You can incubate single cells. It's very easy storage. It's high throughput. It's very diverse applicability, and you can control volume. So you can have different sizes, even down to picoliter size droplets, or bigger if you want. All right, but let's go further here. So droplet-based microfluidics, it enables studying many scenarios. Okay, so usually in our experiments, we have nanoliter size water in all droplets, and they can act as separate miniature environments. So like I said, each droplet is a single test tube. And there you can actually study thousands of unique individual scenarios. So in each droplet, because it's a one kind of environment, you can mix different antibiotic concentration, you can have plastic, you can have no plastic, you have a different concentration of metal. You can mix and match in these droplets as you wish. Okay. So if we look at kind of general how to use droplets, so you can use droplets to study different aspects of antimicrobial resistance this way. Okay. So in a recent review that we published, you can see more detailed about the different pros and cons for using droplets for the antimicrobial resistance research. Okay, but one of the main things is that it can accelerate experimental timescale. So because of the size, especially, you probably also know about droplet digital PCR, which is used a lot for detection of COVID. So the same thing if you want to detect antimicrobial gene, PCR, it's very fast. Okay, you can expand screening, so different antibody combinations, as I also already talked about. You can even merge different droplets together and thus combine different drug combinations. You can also increase sensitivity. Okay, so you can have different concentrations, and because it is such small droplets, you can have easier detection. So let's say you have fluorescence detection, it's easier to detect and faster than if you had to detect fluorescence in a larger volume. Okay. And also it can provide discrimination between diverse subpopulations. Okay, so if you have different populations, some grow fast, some grow slow. On a plate, you don't see these different, especially slow growing bacteria. However, in droplets, because you separate them, so if you have a sample, put them in droplets, you can actually have a better discriminatory populations. Okay, so you can actually see the slow growing bacteria and isolate them. So if we look now into like more specific examples. So these are from 2024, just two very nice examples I wanted to show. These are not done by us, but really nice work. So for example, Samimi, 2024, in their paper, they performed automated multiplex assays. So they had fluorescence dyes, and they made different combinations that enabled them to make 169 different barcodes. So 169 different conditions. Okay, we can see in the fluorescence channel all the colors, while on the right side you can see the bright field. Okay, and then having these different conditions, then they use the bright field imaging for growth quantification. So they had a Python script, and based to the right on the bright field, they could then see during the different conditions whether the bacteria was growing or not, and how it was growing. Okay, so it means it's a good tool for example, screening of different compounds. Another nice example is to use droplets for rapid antibiotic susceptibility testing. So this was also a nice paper by Riti 2024. So what they did is they did a direct cholesterol anti-susceptibility test of clinical gram-negative bacteria in urine samples. So this is kind of schematic how they did. So they had bacteria, they spiked urine samples, okay, and encapsulated that with the Cholesterium bacteria. And within just 2 hours, okay, just 2 hours of growth at 37 degrees, they could then classify the droplets. Either there was growth or no growth, okay? And they could do that by using convolutional neural networking. Okay, so this means they trained AI on different images to detect automatically is there growth or is there not growth. Okay. So overall, including all the preparation, they could then have results in 2 hours and 30 minutes instead of, for example, usually you need minimum 24 hours or longer to actually get results. So they and I agree that this is a nice tool for optimizing treatment of patients infected with multi-drug resistant strains. Okay, so in clinical settings. All right, but let's get back to plastic and let's get back to our platform. So this is what we use in our lab in Tallinn. So we have a flexible and simple experimental setup because we believe this is the best way to spread the droplet technology to make it as simple and available for everybody. Okay. So what we did, we prepared the samples with bacteria, plastic, and antibiotic. We generated droplets with our microfluidic chip. We did overnight incubation. It can be done faster, so we don't need that much time, but for the experimental setup, for the simplicity, we did overnight. And then we did imaging of the droplets on a monolayer, and we did with we used confocal microscope, but you can use any fluorescence microscope you wish. And then we based this growth on either you had homogeneous growth, so it means in the droplet it's quite uniform, the fluorescence of the bacteria, or you could have slightly aggregated growth. You see to the left and down, where some are like more intense pixels and some less. And then either you could have no bacteria or no growth in the droplet, means no fluorescence or just background fluorescence. Yeah. And then we analyzed the images via user-friendly software. So we, again, believe that it should be for everybody. So we didn't want to use any complicated scripting that basically is very unique to each usually group that does it and doesn't work for others, or you need specific training, you need specific things. So we just used free available software. So this is on the internet, Cell Profiler, free to download. It's not ours, okay? And we initially combined different software to analyze images, but now we actually have a new pipeline that's just based on Cell Profiler. Okay, so we import images into Cell Profiler, and then data here there's analyzed and data is exported as CSV. So which is very nice. Yes, somebody.
You can keep going, Simona. I muted.
Yes, it was just mine was muted as well, so I unmuted again.
Sorry.
It's fine. All right, so based on this, you can use a module in Cell Profiler, okay, that's called measure texture, and it measures texture based on pixel intensity variation. So to simplify things, basically it looks at the neighboring pixels and looks, okay, are these pixels the same? So same numbers as you see above, or are they different? Okay, and based on that, it gives a number. And it uses a mathematical model for this. It's very old, from 1973. They used a lot. And the closer to zero, the more homogeneous pixel intensity is, and the more it increases, the more heterogeneous the pixel intensity is. So 0, you have no growth. Something in between, you have homogeneous growth usually, and then heterogeneous, you have a high number. Okay. And then, so this pixel texture, it enabled us to actually classify the droplets in 3 categories. Okay, you could distinguish the pixel heterogeneity inside the droplets based on these values. So if you look to the left, you have texture value of the droplet on the y-axis, and then you just have a droplet number. So this is just how many droplets are identified on the x-axis. And there you can see these nice kind of jumps you have. There are several jumps, so you could have more kind of classifications or categories, but we wanted to make it simple, at least to begin with. Okay, so we based it on no bacteria or no growth if it's less than 52, and homogeneous growth between 52 and 192, and above that it would be then aggregated growth. Okay, and to the right as well in the picture, you can see the outlines, how the droplets are detected, and you can see the blue outlines and the white dots that's the detected plastic that we also put in. So this enabled us to see, okay, are these bacteria, are they more aggregated when plastic is present, or are they not? And also, is their antibiotic susceptibility increasing, decreasing based on plastic? So these are ongoing experiments. Okay, so just like a single trial, and right now we have many different experiments ongoing, and hopefully soon we will also publish these results. It's quite interesting, just to give you a teaser. All right, but so how does the current and future plans look like? Well, for our group, as I explained, we have more elaborate, we have more bacteria experiments, like more bacterial diversity with different species and different plastics, but also we're trying to get more elaborate imaging analysis. So we're trying to go some 3D imaging different types. Okay, and we are, as always, open for collaborations or any ideas you might have from your side as well. And also, one thing I wanted to mention to like what's going on in the world actually, and how important you might then see how important the plastic pollution is that actually already in 2022 the UN kind of agreed. I kind of lost my. Or do
Yeah,
you
we
see?
lost your. Yeah, no, we don't see anymore your presentation.
Yeah, I don't know what the. That's strange.
Now we do.
Yeah.
Yeah.
I'll go to the. Actually, where I was. Almost at the end. All right. Yeah, UN. So we had 175 nations actually gathered, and they agreed on a mandate to create a first of its kind, so really international legally binding instrument to end plastic pollution. Okay. Or of course, it's not always easy to get everybody to agree on this, especially certain countries. So they're still discussing as we speak. Latest was 2024 in December, and they're still trying to see how they can mitigate and kind of make this legally binding document. But yeah, it is being taken serious as well around the world now. So with this, I just want to acknowledge my students, Professor Teller, our funding, and also please you can scan QR codes based on our preprint. It's based on our work that I just presented, or the review. If you're more interested in just antimicrobial resistance, you can check that out as well. And that is it. Thank you for your attention.
Thank you, Simona. Thank you very much. Very nice presentation. I just remind you that the chat is open for your questions. So please, in case you have some questions, comments, or curiosities, just write them in the chat and we will address them at the end. It's now the time of the second speaker, Abidelfatah Nasser from Israel. And the title of his talk is Stream Contamination with Emerging Pathogens and Antibiotic Resistance from Point and Non-Point Pollution Sources. If Simona, can you stop sharing your desktop? Yeah, thank you very much.
Trying to figure.
Okay.
I don't know what how do.
There should be a stop sharing somewhere.
It should.
Yeah.
And I know it's very strange.
I can do it for you if you want,
Yeah,
Simona.
please.
Okay.
It's very strange because I've done this. It usually is a stop sharing, but it's like it's very weird. But thank you.
Okay, thank you.
Okay.
Eva, sorry, just a communication. Now I can see the chat, but I don't see this message sent up to now. So if there are
there
messages...
is one question.
Now I see, yes. If this is the first question, it's fine.
No, there are two questions.
Okay, so in case if you can copy paste, I can see them.
Okay, perfect.
Okay, Abidelfatah, if you can share your screen, thank you.
Wait a minute. How do I share my?
There is a button share with
on the top.
Yeah, on the top
to the
right. It's a box with an arrow pointing up.
Okay, I see it.
And you have to say full screen. You are
Now
muted.
you are muted.
I am okay.
Muted again. You now.
No, now it's working.
No.
Yeah.
I don't see my, yeah, my presentation is here.
Yeah, try to go full screen.
Can you see it now?
Yeah,
Perfect.
try to go to the second slide to see if it is working. Yeah, perfect. So you can start. Thank you very much.
Okay. You're welcome. My presentation is extreme contamination with emerging pathogens and antibiotic resistance from point and non-point pollution sources. This study is part of a big collaboration with the 5 European partners and 2 African partners. It is about the contribution of streams and rivers to the antibiotic resistant contamination of surface waters and oceans, seas and oceans, especially the contribution of wastewater treatment plants to the contamination of surface waters with pathogens and antibiotic resistance. What I'm going to present here are indicators of pathogens, which are bacteria and viruses indicators, and antibiotic resistance based on the ESBL producing bacteria.
I don't know if it is just me, but I don't hear him. Now.
Yeah. Now, yes.
No, I still don't.
No.
No.
He's somehow frozen.
And yeah. Abby, you are having some issue, some connectivity issues, and we
Yes.
hear you. Yeah. You come in and out.
Now?
Okay, now, yeah.
Yeah. You have just to go in presentation mode.
Yes, I will do that. What you can see in this slide are the most abundant bacteria, viruses, and protozoan parasites which are transmitted by water. What I had to say at the beginning that my specialty is health-related water microbiology, which includes all these pathogens, and recently we included in our area the antibiotic resistant genes and bacteria. So most of you know this topic. To those who are not familiar with it, what you can see here are the main mechanisms of antibiotic resistance and how the antibiotic resistance is spread by horizontal gene transfer, which can be very fast, especially after treatment of water or wastewater. We make the bacteria permissible to the genetic material. Most of the studies done in this area, what we did also in our lab is the enhancement of transformation process by chlorination, which is used everywhere in the world. Chlorine for water disinfection. Okay, so this is the antibiotic resistance is a topic which continues with the microplastic, because I think the bacteria with efflux pumps it produces makes them also the exposure makes them also antibiotic resistance. Okay, so just a background for the antibiotic resistance around the world. What you can see here is the map of the world. The light blue is low antibiotic resistance, and the high resistance is the dark blue. The darkest are the most where the most abundance of the antibiotic resistance. Some of the studies trying to do it by other means, like the measures that I gathered from the internet. What you can see here is 3 indicators which can predict the antibiotic resistance, probably. The defined daily dose of antibiotics, the amount of antibiotics used in each country. This is one. The second is the infant mortality rate per 1,000, and the GDP is the how much the people earn in each country. What you can see here, for example, you can see the red ones, okay, where the mortality of the infants is above 4 or around 5 per 1,000, and the income, the DDW is sometimes it's very high, like in Bulgaria, Romania. However, in places like Malta and Slovakia, the DDD is not that high. Okay, so this is something that people can, in some of the reviews I saw it, that people try to explain why the dark blue is abundant in some countries for the antibiotic resistance. Okay, so the aims of our study, for all the partners in this study, we looked for these indicators and pathogens and antibiotic resistance in one stream or river in a country. And we wanted to determine the prevalence of and persistence of indicator bacteria and coliphages, as well antibiotic-resistant bacteria and antibiotic-resistant genes in wastewater and receiving streams. So in terms of wastewater, we wanted to look for the reduction of these parameters in the treatment plants. Therefore, we took wastewater influent and effluent, what you will see in the results. And the common samples that we used were the raw wastewater and after secondary treatment. Okay, we wanted to see how the anthropogenic activity affects the microbial contamination and the receiving surface waters like the sea or the ocean, because all the rivers flow either to the sea or the ocean. And in terms of point and non-point sources of pollution along the rivers, sometimes the natural purification reduces the amount of contamination. However, if there are some non-point pollution, then there is all the time contribution of these non-point contamination sources to the contamination of the rivers and the streams. And this is what we will see along the presentation that I will present here. So in Israel, we concentrated on a stream called the Yarkon stream. it's in the central part of Israel. What you can see here are 5 sampling sites. The origin, which is very clean, as you can see here, and the wastewater treatment plant, influent and effluent, the raw wastewater and the treated effluent. Downstream, it's about 10 kilometers from the wastewater treatment plant, and we wanted to see how contaminated is the outfall to the Mediterranean Sea. Okay, so the treatment plant that we looked for is advanced treatment, in which the wastewater is treated by activated sludge. It's biological activated sludge, which makes it very high in terms of very high quality in terms of suspended solids and organic material. Then there is a treatment process called sand filtration, and after sand filtration, the treatment is either the effluent is either disinfected by irradiation, UV irradiation, and discharged into the stream, or by chlorination and used for unrestricted irrigation. I don't have the time to explain why this practice is used here, but the unrestricted irrigation, the chlorine disinfected is within the standard for unrestricted irrigation all the time. So the parameters that we looked for are in Israel are E. coli, fecal coliform, ESBL E. coli, sulfate reducing Clostridia, somatic coliphages, and then ARGs, and the total bacteria is 16S rRNA gene, and the most abundant genes that we look for in the literature were SOL1, INTL1, and TIT-A. This is for integron class 1 gene. The SOL1 is for sulfonamide, and the TIT-A is for tetracycline. So then I will show you the results. The results look like this. The origin for fecal coliform results in the Yarkon stream, the origin was very clean. The raw wastewater was very contaminated, and then the secondary effluent was reduced by 2 logs, 2 orders of magnitude than the raw wastewater. But however, the downstream were contaminated as much as the secondary effluent. Okay. These are the results of the ESBL E. coli in the stream. In the origin, there were no bacteria detected. The source of the contamination is the raw wastewater. Secondary effluent was by at least 3 logs or more reduced in the secondary treatment. And the ESBL bacteria was also detected in the downstream and the outfall. Okay. The same picture is seen for somatic coliphages. In the origin, it was very clean. Raw wastewater was very high, which is the source of the contamination. And here I have to clarify that somatic coliphages are RNA phages mostly, and they are the indicators for the enteric viruses, which are a huge number of a group of viruses. Therefore, we use indicators for that. What you can see here, in the secondary effluent, 1,000 per 100 milliliter, and in the downstream and outfall, they are still present and persistent. Therefore, we can say that viruses which are present in the secondary effluent and discharged into the stream are also arriving to the outfall. There are two groups of coliphages. One of the is the somatic, and the other one is the F+ coliphages. And some people think that F+ coliphages are more suitable as indicators for enteric viruses. However, here we can see that their level is lower than the somatic coliphages, but they are present. They are not present in the origin, in the upstream, and they are present in all the samples, with highest in the raw wastewater and low. In this case, if we look for the natural purification, it is not working here, because the reduction from the secondary effluent to the downstream and outfall is not that high. It's less than 1 log reduction. So in the secondary effluent, to summarize, in the secondary treatment in the activated sludge, a reduction of the fecal coliform and the coliphages is about 2 logs, 2 orders of magnitude, and the ESBL E. coli is more susceptible to this treatment, and it is reduced by 4 logs. For the coliphages, the picture is different. There are some bacteria in the origin, and there are some antibiotic resistant genes in the origin. Okay. The origin here is extracted from groundwater under the direct influence of surface water. Therefore, some pollution is arriving there, and this means that the result, the PCR, the qPCR system that we are using is very sensitive. Therefore, we can detect SOL1, INTL1, and TET-A also in the origin. The highest levels of the ARGs are in the raw wastewater. However, the ARGs in the other samples, in the secondary effluent, downstream, and in the outfall are very high. Therefore, we can say here that the stream, in this case, is very polluted, and the contribution of the stream to the sea, so the contamination of the sea with ARGs is very high, because this is the outfall, and you can see that the SOL1, INTL1, and the TET-A are very high, higher than in the case of the SOL1 is 8 logs, 8 logs of copy numbers per milliliter, and for the others it's above 4. Okay. The one group of this consortium was responsible for metagenomics, and they got the samples from Sweden, Germany, France, Spain, Portugal, Israel, Uganda, and Mozambique, the 2 African groups, and 5 Europeans and our group. And what you can see is a heat map of 7 ARGs. The highest levels are of SOL1 most of the time. You can see that in some countries it's even higher than the others. For example, if you can look at Sweden, the level of the antibiotic resistant genes is very low. Maybe it correlates with the DDD, the amount of, either with the amount of the antibiotics used in this country, or the amount of rain precipitation in this country, because we look for the ARGs in the surface water and the wastewater. We can see that in Israel, in all the samples, the effluent surface water and the outfall is the soil is very high. Sometimes the tit C, in this case, they did not take the tit A like our study, they took the tit C to present. And you can see that the tit C is, in some countries, is very high, especially in the influent is very high. Okay, so this map, it can be a platform to the review that I propose to work on in our cost action, and some of the participants already expressed willingness to participate, and this is something that we can use and share. In the conclusions, we can say that the levels of the fecal coliform, the indicators, and the bacterial indicators and the coliphages are very high. And this means that there are some non-point pollution sources along the stream. We can say that the ARGs are very persistent and the microbial indicators are very persistent, and they can reach the sea and the ocean. And this may be an introduction for to assess the risk for public health using this beach and the sea for either recreation or fishing. This study was funded by the Ministry of Health in Israel in the frame of the ERANET, which is aquatic pollutants, joined with the 5 European partners and 2 African partners, and the consortium is called ZARA. You can find it in the internet sometimes. Thank you for your attention.
Thank you very much, Abidelfatah, for the very nice presentation. We really appreciate, and we see that some questions are rising in the chat. So we can now move to the last but not the least speaker of today. If you can stop sharing by click on, yeah, that should be the button. Thank you very much.
You're welcome.
So it's now time of Amparo Faustino from Portugal with a presentation, Can Photodynamic Inactivation Control Microbes and Mitigate Resistance in Water? We are eagerly looking for an answer to this nice question.
I tried to, the system said this is blocking screen, share session. Probably I need to have permission to share my screen.
I don't know,
In
Eva,
principle,
can you check?
No, in principle, we all have permission to do it, as the other speakers have been sharing. There is no blocking. Can you try again? Maybe it was because Abid hadn't stopped sharing yet or something like that. Can you check?
I try at least. No, system is blocking. I will go out and restart again. Probably it's easier. Okay.
Yeah. Let's see if it works.
Okay. No.
Alternatively, you can send me the presentation and I will share it from my laptop.
Okay. I think it's better. Okay. No, let me see. Just send that. Yes.
Okay.
Just 1 second. Already sent, but we have a delay of 1 minute. So.
I'm checking.
Probably is already in your site.
Not yet. Not yet. I'm checking.
Because it's a little bit big, so
heavy. Yeah.
Yeah.
Mattia, maybe you want to take this little time to go through the questions while I.
Yeah, why not? We can start. I see Simona is still there, so we start with questions for Simona. Any challenges for sorting targeted droplets for downstream application?
Yeah, I mean, there is a lot of more now of droplet sorting being done. I guess one of the main things is we're also trying to build a custom one for ourselves, trying to get cheaper because the price usually they cost a lot. It's commercial ones, and usually if you sort droplets, unless you can use gel droplets, but if you use water and oil, you better have your own fax, or you better have your own sorter, because there is usually oil residue as well that comes in the machine, and then people get mad at you if others are using it as well. So you can't really use a shared sorter with others that are sorting other cells. So there are still some issues, but it is possible to definitely sort, and people are sorting and doing genomics or sorting different for compounds, definitely. But there are still some technical challenges.
Okay, then Eva, can I go for a second question? Yeah. So when you analyze bacteria cells in fluorescent microscope, do you use any specific dye to color the cells?
So you don't have to. So we also have some strains that don't have any fluorescence. You can just do it in bright field, and you can see whether they grow or not. Just to simplify the analysis, we have strains that have incorporated fluorescence proteins, for example, GFP, E. coli, that in the chromosome. So if they grow, you see fluorescence. If they don't, you don't. So that's the easiest to do, like simple experiments with students, but you don't have to use any fluorescence, whatever, if you don't want to. Yeah.
Okay, thank you. So it seems we have now the presentation from Amparo Faustino, so we can stop the Q&A session and we will resume it later after this talk. You are muted, Eva. again. Eva, we cannot hear.
Okay, I'm just trying
Okay.
to make. Just give me a minute. Can you see it now?
Yeah, we can see.
So, Amparo.
Okay, we can start.
Stage in
Amparo,
Spanish.
you can start your talk, and you may simply say next when you want to have a change of slide. We cannot hear you. Oh, yeah, because she was not connected anymore.
With my
Yeah, we can see you now.
I lost the video, the ability to talk. So
Your presentation
eventually,
is loaded. Can you see it?
yes. Okay.
Okay,
So,
you just say next when you want to have a change of slide.
or eventually, I think now I can also let me see, share, because I switch off completely the system, and now I think, can you see? Yeah.
Yes.
Okay. I
Yes.
think now I
Yeah.
can. I think it's better than to say something
Yeah.
like. Okay. So.
Thank you. You can go. Thank you very much.
Sorry. Sorry. Thanks once again for your invitation, and it's a pleasure to me to be here and to share, to contribute also, because in fact these two actions are complementary. So my talk today is about if the photodynamic therapy inactivate and control microbes in water. And for that, I will briefly introduce photodynamic inactivation. To do this approach, we need three components. The photosensitizer, a drug, or at least a dye compound, oxygen, and light, and namely visible light, not the non-ionized radiation. And what's happened? The combination of these three components produce the cytotoxic species, namely singlet oxygen. So the toxic molecule are not the molecular oxygen, not the light, nor the photosensitizer, the molecule. And how this occurs? This occurs based on the compound absorb light, became in the excited state, and then through interstitial crossing process pass for a triplet excited state, and then can by transfer, electron transfer or energy transfer, to produce reactive oxygen species or radical species or high reactive dioxygen. And are these molecules the responsible for the inactivation of the microbial cells? That means the photosensitizer acts as a catalyst. So one molecule are able to produce several reactive oxygen species. And because this is not a target approach, so all the compounds, the components able to be oxidized are the target of this approach. And the benefit is this method can work in sensitive and resistant strains. And it's very, very a good point. Moreover, in the past, when the photodynamic therapy starts, especially for antitumoral approaches, they require very specific light sources. But nowadays, we can use fluorescence bulbs, halogen bulbs, LEDs. So it's very easy to use in several for several applications, and the cost is reduced dramatically if we compare the price of a LED compared with the laser light source. So, thinking in this approach, we could, with last 20 years in our group, we try to use it as antimicrobial tools. And we start to think, can we use instead artificial light? Can we use solar light to inactivate, to produce these reactive oxygen species? And what we performed that time was using a gram-negative bacteria, transformed bioluminescent strain. We test, because we are a synthetic group working in porphyrins, we prepare tetracationic porphyrin derivatives in different concentrations, and we select artificial light and also solar light, removing the UV part of this light. And we perform our experiment and in parallel, and what we observed was that when we use artificial light, in this case was bulbs, 13 bulbs, fluorescent bulbs, what we observed was we was able to inactivate the gram-negative bacteria, E. coli, and more than 99.9% after with 5 micromolars. And after 217 minutes, we completely inactivate we reach the limit of detection of the method. As you can see, the photosensitizer without light is not toxic, and the light used also the black line means that the light alone do not produce any inactivation, do not inactivate the bacteria. When we use light, the solar light, what we observe? We observe in the 3 concentration tests, we reach the limit of detection of the method, although the light will remove the UV part of the light, and as you can see here, during the experiment, we are not able, only with light, solar light, to kill this E. coli. So the first thing that we can say is we can use even not only artificial light, but also to use solar light to promote the photodynamic inactivation of bacteria, and especially in not in the north, but in the countries of the south and Mediterranean countries in Africa, we can use it as our light source. Second thing is, can this approach work in gram-negative in resistant clinical resistant bacteria, and for that we test E. coli for bacteria, for resistant bacteria collected from the hospital of Coimbra, and we use the same photosensitizer, and we test 5 micromolar, and in this case we test with the light, the artificial light. For the approach was the same, and what we observed was for the 4 bacteria, 2 positive bacteria, gram-positive bacteria, and 2 gram-negative bacteria, what we observe, it's in all cases, we are able, even these bacteria were resistant to antibiotics, we are able, with different profile, but we are able to inactivate this bacteria with this porphyrin, tetracationic porphyrin, after 10 minutes of contact, and then expose to light. But these bacteria are antibiotic resistance, and what's happened to this if we combine the porphyrin, the photodynamic approach, with antibiotic approach? What's happened? And in this case, we test Escherichia coli resistant to antibiotics, and we expose this bacteria to our porphyrin derivative, and exposed to light in the presence and in the absence of antibiotic at the MIC concentration. And what we observe, so we test the photosensitizer alone, photosensitizer with antibiotic in this, and with the light only with antibiotic. So all these 3 setup were irradiated, and here we use the dark control using without just photosensitizer and antibiotic without light. And what we observed, as you can see here, using the MIC concentration, we are able, without antibiotic, reduce until 6 logs of at the end of the experiment, 6 log the load of the bacteria. However, in the presence of the antibiotic, we improve this inactivation until the limit of detection of the method. Almost 8 logs of reduction was observed. It's interesting to note that the dark control, so the porphyrin and the antibiotic together without light, do not produce any inactivation. Even the light, the antibiotic in the presence of light, is our light control, is bacteria, antibiotic, and light, we cannot observe any inactivation. That means the combined action of the porphyrin with the antibiotic improve the inactivation, the ability to kill these resistant bacteria. I remember this bacteria is resistant to this antibiotic. So our second remark means we are able to efficiently inactivate multi-resistant bacteria with this approach. We can do it in a micromolar concentration. In this case, we also do not only inactivate the bacteria, but also overcome the problem related to the bacterial resistance. And the combined action of the antibiotic and the photosensitizer is more efficient than each approach alone. But can we, this happen in PBS, in the control microcosm, but can we do the inactivation in real wastewaters? And we perform the same experiment using the wastewater from the hospital. So we collect in in 3 different days wastewater from the wastewater plant in the hospital. We we filter this water to remove the other bacteria and put inside add our bacteria to control, to verify. And what we observe for all the 4 the resistant bacteria strains that we test before, and although the profile are different depending of the day of we collect the wastewater, and in all cases we were able to inactivate all the bacteria in different times, but we was able to inactivate this bacteria. What this means? Yes, we can, using real wastewaters, we can inactivate the multi-resistant bacteria, and this open the possibility to use this approach to include it as a novel or another approach to wastewater treatment, especially this specific high-loaded antimicrobial, high-loaded microbial wastewaters. But okay, that water in the hospital have a lot of antibiotics, detergents, so it's like we have the combination between the photosensitizer, the photodynamic inactivation, plus the antibiotic dissolved in this hospital wastewater. But what happened with if we consider to treat different wastewaters from different sources? Can we also mitigate this? And as I can see, we test it. We use 9 different wastewater origins. And as you can see, domestic, industrial, seawater, water from aquacultures, pounds, and what in this case we don't use we use not a single photosensitizer, but a formulation prepared by us in our lab. We use as our model Escherichia coli, and also we use the ELAD system. And just to show, I would like okay, it's a very short, small data, but just to look the profile. Different water collected in different days. In all cases, we were able to inactivate the bacteria in all these in all these wastewaters. Moreover, we also test. It's not I don't have time to explain, but we try to identify the parameters responsible for these different profiles. But the idea that I would like today to pass for the audience is, yes, it is possible to inactivate our waste, to use this approach to photoinactivate bacteria, but also today I show only bacteria, but we can also do it for virus, for instance. Okay, So in this case, in all cases, after 61 hour, we are able to inactivate 99.9% at least of bacteria. But, and if you will repeat this approach several times, what's happened? Can we develop, we know the bacteria develop some resistance to this treatment? We also address this point, and what we observe, we use one of our porphyrin, cationic porphyrins, and as you can see here, we have complete inactivation with this bacteria. We observe very fast inactivation decay, and because we thought with this kind of inactivation, if the bacteria, it's a bioluminescent bacteria, if this bacteria could recover the bioluminescence or eventually be dormant and then be reactivated. In fact, we observe after 1 week the bacteria still it's not, and we also count the colony form units after the 1 week, and no bacteria remain. And we try to use that this time. It's a sub-lethal, so before we kill all bacteria, we collect the remainder survival bacteria, and we promote the regrowth of these bacteria and test again several times, 10 times. And what we observe, we use 2 kinds of bacteria, Escherichia coli and E. coli, the 2 bioluminescent, one natural bioluminescent bacteria, another one transformed bacteria. And as we can see, we don't have significant variation after 10 treatments related with the sensitivity to the photodynamic approach. What I want to say is we can achieve inactivation in several water matrices. So this approach can be effective to inactivate the bacteria. This technology can consider a suitable sanitation tool, and no resistance mechanisms we can detect in bacteria after consecutive generation of surviving bacteria. If you want to know a little bit more about this last study that I show, you can see the literature. It's already published, and I finished as I started. Can the photodynamic inactivation control microbes and mitigate resistance in water? The answer is yes, we can. Finally, I would like to advertise you for the next Congress of the European Society for Photobiology in Bari, where these and other topics will be also addressed. All of you are welcome. And of course, this needs people to do that, and it's only possible due to the fantastic team that is behind me, or not behind, in my side, to do all this fantastic work. And thank you.
Thank you very much, Amparo. Very nice talk. We really appreciate it. So if you can stop sharing, I would like to thank all the speakers. Very nice talks. We have a lot of feedback in the chat, and I would like also to thank all the participants for the keeping active the participation from the beginning to now. So we can go ahead with the questions. We were stopped at this question from my side for Simona. I was curious about the addition of plastic into your protocol. What can you say about the plastic? Is a standard operating procedure, or you need to have a specific plastic type, or you can change it? And in case you can change, if you have some difference in the outcome of your assay based on the plastic type and features that you add?
It's a very good question, and I would say the more I get into micro and nanoplastic research, the more standardization comes into play as a crucial aspect, because there is a high lack of standardization. It's so hard to standardize anything in the micro nanoplastic research, and that's one of the main problems, is actually that standardization of methods and having proper reference material. Because, for example, we use, as I can say, 98% of research right now, basically, or 99, I would say. For reference material, the type of plastic is polystyrene, and it's not the most abundant one.
Please mute your microphone in case. Thank you.
Merhaba.
Thank you. Yeah, it's not the most abundant one out there. However, it's so hard to get actually reference material commercially, or it's a pain in the butt to make it yourself, and you can't really even like make it then like equal sizes. Yeah, so it's a whole different problem to do so actually. I can say we use what's commercially available, and but there's a huge discussion currently in the microplastic research community. We need better standardization overall in these experiments, and I'm also in the cost action for that priority, which deals a lot with that as well. Yeah.
Okay, thank you. Another question, still for you. Which antibiotics did you test in your assays, and have you found a pattern in some specific antibiotics class and increased resistance in the presence of plastic, or is it a general trend?
It's not a general trend. It varies with antibiotics. We have different tests. I mean, we have, for example, ciprofloxacin, we have canyamycin, we have cefotaxime. I think we added as well. Yeah, we had, I can't remember exactly, like we had now many different ones that we are testing currently with students. There's no right now specific trend to what mechanism they target, like then this matters how the antibiotic is susceptibility is affected. However, we're still actually doing a lot of the analysis as we speak, so maybe we will find a trend. So we are doing because we're waiting on some software things to because things constantly develop. So now we have now is the time to so I have to keep you waiting on the specifics on that. But you're most welcome to contact me anytime as well. We'll gladly give more details. Hopefully, maybe even next week I will have. Yeah.
Okay, thank you very much for the fresh update. Now we move to questions for Abidelfatah. The first one is, while investigating bacteria contamination, did you guys also check for the presence of antibiotic in the stream?
No, we did not look for antibiotic residuals in the stream. Maybe there are some other groups who looked for that. We looked only for bacteria, ESBL, E. coli, and antibiotic resistant genes. This was our aims.
Okay, thank you.
Maybe there is a correlation between them, between the residuals and the antibiotic resistance, because this is the selection, maybe.
Yeah, probably it's related also to the next question, because it asks why the 16S copy number did not seem to reduce across the wastewater treatment. Could you explain why?
A good question. We look for the DNA. We don't look, when we detect the 16S, we don't look for the whole bacterial cell. The whole bacterial cell was reduced by 2 orders of magnitude, and the 16S is only DNA, and we look for a short fragment of the DNA. Therefore, maybe the reduction here is not very efficient. And the secondary treatment mostly used to reduce organic material, assimilable organic material, which is decaying very fast, and the suspended solids, not for bacteria or viruses. The reduction of bacteria, viruses, and DNA is very inefficient in the secondary treatment.
Okay. Okay, thank you very much. We now move to the questions for Amparo. There was a problem with displaying the presentation, but we hope it was fixed. There is a question from my side related to the first approach that you showed, not the one with the combination of antibiotic and photodynamic. You mentioned the dark control. I got a partial answer during your talk, and just asking if the dark control was including the TMPY. Yeah.
Yeah. We always put the dark control is the photosensitizer at the highest concentration tested, because if it's not happen nothing, that means below doesn't happen. So always in our experiments we use the light control, just bacteria and light, or bacteria, light, and the other additives that we put for the PDT assay. And for the dark control, we also tested everything that we put in the PDT or PDI assay, less light. So the dark always have bacteria, the drug, the antibiotic, whatever, and always at the highest concentration.
Okay, thank you very much. Now, question: using your PDI approach to inactivate bacteria, do you also check the concentration of ROS in the treated water? Given the negative effect of ROS in human and animals, how can you mitigate the risk of this ROS produced via this approach?
The main reactive oxygen species using porphyrins is the singlet oxygen, and the singlet oxygen, the action, so it's less than 1 micrometer. That means it's localized action, and when you remove the light or the photosensitizer, disappear the effect. So the effect is very localized. We have another recent work where we verify the effect when we release this water. For instance, the wastewater treated with these porphyrins are released in the water bodies. What's happened? And the effect of the dilution. And we observe if you diluted 1 to 50%, we have affecting the native bacteria and in the environment. But we don't dilute only 2. The dilution is higher. For instance, 110, after 110, we don't have any effect in the native bacteria in the environment. Even so, we don't want to replace biological contamination by a chemical contamination. And the next step, and we try to work on this, is to immobilize these photosensitizers. We have already for the next webinar, who knows? We have also some data using immobilized photosensitizers, even in films, in ketosane, but in silica nanoparticles, magnetic nanoparticles, that we introduce in the pools, for instance, and then after the treatment, we retain it physical or in general physically, because the compounds are connected, are linked to a support, insoluble support, in order to not contaminate the environment with this chemically with this photosensitizer to avoid the bioaccumulation and so on.
Yeah, there is a question. I jump and then I come back because there is a question from Vincenzo Summa, which is related to this answer that you gave, which is the concentration of PDI is high and you have to consider the impact in the natural environment. And then there is a suggestion for using immobilizing matrices to separate by filtration and recover. So if you can add, if there is any detail that you want to add to this question as well, or if Vincenzo want to comment.
We have some. We already mobilized in Merrifield or in silica nanoparticles or in magnetic silica nanoparticles and in films, in different in natural films. So we already, especially with Merrifield, for instance, we are able to apply several times without loss of any activity. With the silica nanoparticles, magnetic nanoparticles, we had some. We lose some activity after a few cycles, but we try to fix that. So it's the research. So step by step. But it's the idea is, if someone has some other ideas, we are open to discuss and to join our molecules. For instance, I mentioned in the last, instead to use a single molecule, we can use a formulation that we can immobilize in these supports in order to reduce the costs and improve the efficiency.
Okay, thank you. We move to the next question, which is quite interesting because they ask for the challenge that you see with your approach working in natural system, like, for example, wastewater with soil contamination or in setting tanks. This will be very relevant for pathogens and AMR reduction in vegetable production in Ghana, but also in other countries where they make large use of wastewater for irrigation.
I think what we perform, because we need to have some control, otherwise we cannot understand the data. We remove, we filter we perform 2 kinds of experiments. One is we collect the water, so all the chemical components inside is still there. We just remove the solids, the suspended solids, and the bacteria. Otherwise, we cannot know because we cannot. I'm a chemist, not a biologist. So forgive me if I don't use the proper words, but we remove the main bacteria and in order to have a control system. Otherwise, it's impossible. Because if do not work in the control system, for sure will not work. Probably it's impossible to control in the real wastewater with everything. Even so, so the first part we remove the solids and the solids and the bacteria inside them. So we filter. But all the other chemicals, the detergents, the antibiotics, the heavy metals, if it's industrial, depends of the kind of an industry we will have. All for the aquaculture waters, we have also some a lot of organic compounds there, and all of these things affect but even so, we are able to, sometimes what we need is to increase the amount of the photosensitizers. But we can also, but if we immobilize, no problem, because although we put more, we can recover at the end of the system. And this could be very interesting, especially in system of circulate, not in the open field, but in system that we have the circulated water. Okay, at least for now.
Yeah.
And of course, after, if you remove, you can irrigate, use this water to irrigate. It's our idea because we need, the water is the more precious thing that we need to keep safe. So we need to reuse the water. Otherwise, we don't have water, fresh water in the one decade.
Okay, so we have now the last question. Before PDI treatment of any type of water, do you first identify the bacteria species contaminant in each of these water sources?
We have an idea about the bacteria because when we, especially in the results that we did not show today, we verify what kind of bacteria that we have, or at least the families, because many of the, as my colleagues from biology department say, sometimes we cannot, not all the bacteria present in the water we can grow. We have conditions to can identify. It's why we selected, in general, why we use E. coli or these bioluminescent E. coli. Because first, because you can immediately, it's very grateful for the students. After the experiment, you have immediately the result. Even if you count the colonies, after you perform your assay, and then you put in the luminometer to collect the emission light, and you have an idea about if the system work or not. It's the first thing. Second one, it's other. If work your photosensitizer works in PDI for a gram-negative bacteria, for sure you need, if you need 1 hour to kill the gram-negative bacteria, you need, with the same concentration, the same light dose, you need 5 minutes to kill a gram-positive bacteria, even a multi-resistant Staphylococcus aureus. Okay, so it's why we use And it's why we are confident if this approach works with these gram-negative bacteria, will work with gram-positive bacteria, with fungi, with in biofilms. It's challenging also, but it's possible. And the idea is not to destroy the biofilms, but avoid that could be installed in the system.
Thank you very much. Abid, you raise your hand. It was a question or just a side
I have a comment.
Yeah.
You said that you are not a biologist, and I am a health-related water microbiologist. So I want to comment about your approach. E. coli is used as the microbial indicator for water quality, okay? But it is not the most resistant microbe in water, especially if we want to test inactivation of microbes in water to see that they are safe for irrigation and reuse. We need to check for viruses, protozoa, and so on, because they are the disease-causing agents. So your approach is fantastic, but you need to widen the parameters that you are testing. For example, to include spores or include some viruses, but your approach is perfect.
Thanks very much for your comments. You are right. We have also data with the virus, and it's easier with this approach to kill virus. It's easier than to kill bacteria. The protozoa is a little bit different. We don't have. I know some groups in Algeria works with Giulio Iori in the past. From the University of Padova, and have had some results and were able to kill to destroy helminths. And related with the virus, the spores, it's a challenge to kill spores, but it's possible also. We have some in our group, some data in the past when they started to the big issue about the anthrax. We try, and the group of Michael Amblin in Harvard tried to test the to destroy spores based on the photodynamic approach, and at that time we, almost in the same period, he test and we test in our lab, and he say, oh no, the porphyrins didn't work to kill spores. And surprisingly, we have results with porphyrins, good results. In fact, then we observed that the porphyrin that he used was not really good. We synthesize the porphyrin and we test with our, because we say, probably we missed something. And we perform our protocol with the porphyrin that Amblin tested and didn't work. But that means we are able also with this approach, depending on the porphyrins, of course, but we are able to kill, to destroy also spores. But you are right. It's not this. We need to see it like a whole, not a piece of. Today I only show a piece of this big world that is the microbials in water, wastewater.
So we are looking forward to see the other pieces in the next
Okay,
event.
thank you.
Okay, so we can close the question and answer session. Eva, do you want to close the webinar?
Sure. I just want to say thank you so much for the speakers, the questions, the discussion, the contents, and the promise to be able to learn more. Thank you, Mattia. Thank you to everybody, and hope to see you soon.
Thank you very much.
Thank you. Thank you once again.
Goodbye. Thank you very much.
Thank
Have
you.
We
a
will
nice
share.
lunch.
Bye.
We will
See
share
you.
the recording. We will share
Yes.
the recording.
Okay,
Thanks.
thank you.
Bye bye. See you soon.
Bye.
Bye.
Yeah.
Bye. See you soon.
Bye bye.